So we're here at the Biologists at 100 conference with Jenny Richens, and you have a preprint out at the moment, and it's been accepted?
I do. It has been accepted.
Congratulations.
Thank you very much.
So I was just going to ask you a few questions about your preprint. So
Yep.
first of all, could you tell me in about a minute roughly what your preprint is about?
Okay, so my preprint, well, mine and my other two co-first authors, is about a tool that we have developed to enable us to do tracking in low signal to noise environments. So the environment in particular we've been working on is the follicle epithelium of the Drosophila egg chamber, where normal tracking tools that were already out there, you just can't see what you're trying to see, and manually tracking vesicles moving throughout the cell would be a nightmare. So I, co-author Maria, wrote two pieces of software, one of which actually detects the tracks and links them into, well, detects particles and links them into tracks. And then the other is a viewer software, MSP Viewer, that allows you to import the tracks from MSP Tracker, the software she wrote, or from other tracking software if you just like the viewer, because it's a really nice piece of software. And then you can curate your tracks, add any tracks that the tracker missed, delete any where you're like, that one doesn't make sense. And so that's kind of the software side of the paper. And then the biological key take-home messages are that we can use the rush system in flies to trap our cargoes and then watch a wave of secretion go through the follicle cells. And we looked at two different cargoes. One was Cadherin 99C, which is an apical microvilla protein, and there we showed that it is transported on microtubules by dynein basically straight apically in the cells. And then the other cargo we looked at is nidogen, which is a soluble ECM protein. And that, while we didn't see a bias in its apicobasal trafficking, even though it comes out at the bottom of lateral, we did see that along the base of the cell, it is planar polarized towards the leading edge of the follicle cells when the whole egg chamber is kind of doing a collective migration.
Rotating
early
around.
and yes, it's rotating. Yes, this is actually really interesting because this is the opposite of what has been seen for collagen secretion. Oh. So that seems to be biased towards the trailing edge of the cells. We have no idea why this would be, but
it's a fun question.
Yeah. Maybe we will find out at some point in the future.
Yeah. And so with getting this work to the preprint stage, what did you find to be the most challenging aspect of that?
I think the hardest thing really was trying to work out how best to balance the biological findings that we had with the software development and how to use the software. And because you can't have a manuscript that is the size of the world. So you've got to kind of squish it down somehow. So in the end, we kind of emphasized the biology in the paper, and then the results that we got using the software. And we do have a description of how the software works and everything and what maths it's based on and all that. But then we've also separately in a repository provided extensive documentation and little videos of me using the software to show people how to do it. And there's a manual of how to go about tracking your things. So we've got all of that kind of elsewhere. So hopefully everything is.
I think that kind of thing is really useful because you have those softwares and then you have no idea how to use them. So actually having that kind of repository will help plenty of other people who
hopefully. I mean, I think it should be a really useful tool for, it doesn't need to just be Drosophila follicle cells. I think it would work in all sorts of different situations where you've got low signal to noise and other tracking software isn't kind of performing for you.
And then, so this preprint was out and now accepted, but in the interlude, did the preprint benefit you personally or the group in any way?
Yes, so the preprint has benefited me because, as you said, it's been accepted now as the paper, but it's not out yet because we have the preprint. Somebody has cited the preprint, which would not have been possible had we still just been in the peer review cycle. So that was.
So somebody's been. It's been useful for somebody else.
It has been useful for somebody else. Yes. Yeah. Which is really nice to see, and
and I guess we just kind of touched on it, but then What do you think the importance are in general for preprints in the publishing landscape and in the scientific community?
So I think they're really, really helpful because you get to have an idea of what's going on in your field kind of much more as it's happening than you would if you were waiting for papers to have been, I mean, peer review, very, very important, but it means you can see what's going on a lot in a lot more timely manner and get ideas that can help your research kind of sooner than you would otherwise, and it can help you to direct your research in a more maybe efficient and more exciting way.
Yeah, nice. Okay, well, thank you very much for agreeing to do the interview.
Thank you for having me.
We'll put the link to the preprint in the comments, and maybe there'll also be a new link for the final version as well.
Fingers crossed.
But yeah, we'll certainly have the preprint link in the comments. And thank you for coming, and I hope you enjoy the rest of the conference.
Yes, it's been fantastic so far.
Perfect.